Okay, time to start. Welcome everybody to this seminar in Ecology and Evolution of Montpellier. For those of you who are online, as usual, we are in a webinar mode, which means that your camera and mics are automatically switched off. If you have any technical issue, please use the chat to report. We will try to help you solve it. And otherwise, if you have questions to today's speaker at the end of the talk, you can use the Q&A tab to ask your question, and then we will allow you to open your mic to ask your question directly. And with this, I give the floor to Michael to introduce our speaker.
Hello, good morning. I'd like to start by thanking Delphine for co-hosting Jens over these 2 days. Unfortunately, his trip has been very short, and hopefully in the future he'll be able to come back for a longer period of time and meet all of you. Thinking about Jens, I first met him in 1998 at a host parasite conference in Banyuls. And the only thing I remember about that conference, aside from meeting interesting, nice people, but no real details about them, was being on the side of the shore, drinking Banyuls with everybody. So perhaps Jens has more memories about these drinks that we had during this workshop. And we all kind of remember there's this group shot photo that everyone was provided with. And I think most of the participants maybe lost theirs. I actually hung on to mine and sent it to Jens a couple of years ago. And he was, oh yeah, there I am. And it's like in the back. And he was a PhD student at the time. So this is 1998. He completed his PhD in 1999 at the Braunschweig in Germany. He'd done postdocs, notably at the Center for Population Biology at Silhouette Park. He had a Marie Curie Research Fellowship at the time, and then became a lecturer and then a senior lecturer at the University of Sheffield, where he was for a number of years, and finally moved in 2012 to the Freie Universität in Berlin, where he's now a professor. He's had numerous research grants, including at ERC, and his research, aside from what he'll be presenting today, includes immune defenses, bacterial resistance to antimicrobials, and metamorphosis in insects. So a very diverse background, mostly focused on insects. So what I think is actually particularly interesting in what Jens does is his focus on antimicrobial peptides, and this, many of you will know, are particular classes of antimicrobials which hold the promise of avoiding some of the problems that we have with the more classical antibiotics, notably resistance and multiple resistances. One of the dangers of antimicrobial peptides, which are coming naturally from many metazoan species, is that should their use be very intense and resistance involved, that could pose a danger for the species from which they came from. So without further ado, I'll let Jens give his talk. Today, the title is Antimicrobial Resistance Evolution, Antimicrobial Peptides and the Merit of In Vitro Data. Jens.
Okay, well, thank you very much, Michael and Delphine, for having me here. So yeah, I want to talk about antimicrobial peptides and resistance evolution. So the latter part is actually mostly about antibiotics. It's an unpublished study, which we did actually to overcome criticism everybody who does in vitro experimental evolution, because one common criticism is it doesn't tell us anything about what happens out there, in medicine, in agriculture and veterinary science. It's unrelated to that. And I think this will be my final data slide. I think I can show you that it's actually not unrelated, then actually generating quite useful data. But to start with, so antimicrobial resistance is a major global health problem. This is a recent paper published in The Lancet, and it shows in the light blue bars global death within a year of people who were infected and where the death was associated with antimicrobial resistance. And the dark blue ones is actually where people are certain that the antimicrobial resistant bug did actually kill people. And as you can see, this is, I mean, it's very small writing, the screen is not very big, but you can see there's Escherichia coli, Staphylococci, Klebsiella, so all the most important escape pathogens as defined by the World Health Organization, and quite a number of others. And this picture, unfortunately, is predicted to be increasing simply because we use more antibiotics, we use antibiotics in agriculture and veterinary science, and there are all sorts of issues one can do about their use. And when we look into the preclinical pipeline published by the World Health Organization, we see that new classes come up. And especially here, one is interesting, so these are peptides, direct-acting peptides, and these are basically antimicrobial peptides. So antimicrobial peptides, you know, if you have looked at that list 15 years ago, there wasn't a single antimicrobial peptide on there, and now they're actually the second most abundant class under development. So because of this, you know, desperate search for new antimicrobials, people turn to antimicrobial peptides. One other thing which I think is very important is that if you look here, kind of purple slice, combinations are now about 18%. And there is a belief now across the community of people working on antimicrobial resistance that if you use combination therapies, that reduces the probability of resistance evolution. And it's commonplace in viral infections to use combinations. Treating tuberculosis is often done with combinations, but in most cases we use single drugs, but it's coming. And one thing I want to point out, it's not on this list, but colistin, which in the literature is often described as an antibiotic, actually is an antimicrobial peptide and has been around for 60 years. So it was introduced to clinical use in 1961, I think. And interestingly enough, resistance was only discovered and described in 2015. That's a very long period. So for most antibiotics, as far as we know, it takes between 2 and 4 years from the start of usage in a clinic to actual resistance causing an issue. And so that's kind of, you know, the only data point we have from the wild. But it's at least one which goes in a promising direction. So what I want to do today in my talk, I want to talk about 3 aspects: resistance evolution against antimicrobial peptides, and why the probability of resistance evolution against antimicrobial peptides is actually much lower than against conventional antibiotics. Then I will have a shorter part on the response to sublethal concentrations of antimicrobial peptides and why it's important and why that might be interesting to study. And then the last one is about forecasting resistance and whether we can use in vitro experiments to contribute to that idea. And that also goes back to a paper which I will show later, which I wrote with Michael when he was on his COVID-interrupted sabbatical in Berlin. So we started that for, you know, discussing it, and then 2 years later we actually could do it. Okay, but before I start, I just give you a bit of background on what antimicrobial peptides are, because I think probably most people are not that familiar with that. So the first antimicrobial peptide which was properly described in a multicellular organism is cecropin. This is a paper from 1981 from Hans Buhmann's group in Stockholm. And that was this cecropin. It's named after Cecropia, which is a moth. So it was discovered in an insect. And Hans Buhmann himself had published a paper 10 years earlier showing actually that insects have immune defense. This might sound silly today, but back then in the day, people thought insects have no immune defense. Actually, they thought only vertebrates have immune defense. And that was because the approach was to look what we know from the vertebrates, which was mostly studies into cellular defenses and, you know, acquired immunity. So insects and other organisms don't have them, so they don't have immune defenses. You know, with benefit of hindsight, we know that that's wrong. And so most antimicrobial peptides possess 6 to 50 amino acid residues. Some of them are much bigger, like the ones that Delphine works on. They're cationic, so they interact just by the charge with the cell membrane of bacteria, and they're selective, so they don't go for eukaryotic cell membranes or cell surface, so they really go for bacterial cell membranes. And they have different mechanisms under different conditions, which I will show. And just to say, the human AMPs were then discovered about 5 to 10 years later. And then this field really took off of studying antimicrobial peptides. And just, in natural situations, antimicrobial peptides have two main functions. So the name originally was obviously they're killers, and they're very efficient killers. I will show some data, but you need to know that in antimicrobial peptide, they kill within minutes. Other antibiotics usually kill within hours, and they kill within minutes. And they also have another function, they are producing symbionts, whether it's in the human gut, where we have a layer of mucus between actual bacteria and the gut wall, which is also rich in antimicrobial peptides. We have lots of examples in insects where they express particular antimicrobial peptides to actually control their co-evolved symbionts. So there they play an important role, but we are not talking about this today. And if we talk about their killing mechanisms, there are two ways. There are antimicrobial peptides that actually kill with intracellular mechanisms, so they have to go through the cell membrane. But the majority of antimicrobial peptides that has been studied actually goes for the membrane. And there are different mechanisms of destroying the membrane. They can just get stuck in the membrane, or they can punch holes in different ways. So you need a certain concentration of antimicrobial peptides. They attach because of their charge. The membrane gets destabilized and the cell leaks out. And when the cell actually leaks out, that is not very good for the antimicrobial peptides, because then proteases come out and attack the antimicrobial peptides. And I'll show you the effect of that a bit later. And so as of April this year, these are all antimicrobial peptides that are described so far. This is from the antimicrobial peptide database kept by Gus Wang in Nebraska. And you can see the vast majority are from amphibia. And I think that has historical reasons, which go down to one researcher in the US in the 1980s. Quite a lot of them are from bacteria, which is a dark blue bar. And then you have fish, humans, insects, and plants. So the only species which is, highlighted here are humans, Homo sapiens. We have 154 described antimicrobial peptides so far, which is a huge number. The amphibia, so in certain frogs, we have quite high numbers as well. In insects, we usually are between 15 and 30 different antimicrobial peptides an individual can actually express under different conditions. And from all these groups, antimicrobial peptides are under development as drugs, including human antimicrobial peptides, and that refers to the problem Michael addressed earlier. I don't have any data on that, we've done work, but I can allude to that in the discussion. So in one paper which was very influential, a review paper of this one here by Michael Zaslow about antimicrobial peptides of multicellular organisms, which was published almost 25 years ago. It's beautifully written, and Michael has a very beautiful language and is very descriptive. The insects flourishes without lymphocytes and so forth, which is true. But in that paper, he made a statement that despite their ancient lineage, so all three domains of life actually have antimicrobial peptides, archaea, bacteria, and eukaryotes. AMPs have remained effective defensive weapons, and that's because AMPs target an Achilles heel of the bacteria. So thinking about the membrane, and that the membrane can only be changed to a certain degree. Michael was then contacted by an evolutionary microbiologist, Graham Bell, who said, I bet you that they can evolve resistance. So they teamed up, and together with a back then master student, Gabriel Perron, who's now running his own lab, they did an experiment and evolved resistant against Michael's pexigenin, which is a peptide he developed in the 1980s and tried to market as a new antimicrobial drug. And so there are still people who cite Michael's paper to say that no resistance could ever evolve against antimicrobial peptides, which I think is interesting given that he himself has been involved in showing that not to be true. But as I will show you during my talk, the probability of resistance evolution against AMPs is very low compared to antibiotics, and that's good news. And I think it would be very hard to have any drug against which resistance can't evolve, which doesn't harm the host, so the human host or an animal host. Good. So I will go into the first kind of part of data. And I start with conceptual background. So at the start, I showed you this list of collaborators. So because it's not all my own work, and a lot of work I did with Roland Riegesch from the ETH Zurich, which I believe quite a lot of people in the room know. And so we use a pharmacodynamic model as a baseline, which Roland developed with Bruce Levin 20 years ago. And what you can see here, and I just use the mouse so that people online can see it, we have the drug concentration here as in terms of the MIC. Now the MIC is a microbial measure, it's the minimum inhibitory concentration. And the way it's done is you use a multi-well plate, you put up a concentration gradient, you put your bacteria in, and then you check by eye where you can't see any growth anymore. And this is standardized worldwide and has been a standard for 80 years. And it's a standardized medium, it's a standardized inoculum. And it's great that it's standardized because it can be compared. The problem is that a lot of drugs actually don't work ideally in the broth people use, right? And so there has been a lot of criticism of that. And it hasn't been replaced because people say the value of having actually a normed approach to measure antimicrobial activity is more important than actually applying it to the particular drug, which I think is debatable, but anyway. So that's the MIC, and so we have drug concentrations above the MIC and below. And here we have the bacterial growth rate. And that can be negative, so that means because it's a population measure, more cells die than new cells survive. And what you can see here is, so a typical pharmacodynamic curve would look like that. You increase the concentration, nothing happens, and then they start dying, and at the end, everybody's dead. And that can take different shapes. And that will be important in a minute. And from that, you can derive a concept of the mutant selection window. So that's the area of concentration where a resistant mutant enjoys a benefit compared to wild type. And so this would be here the wild type curve, the pink one, and the dashed one is a resistant mutant. And we assume, and that's true often, but not always, that being resistant against an antimicrobial comes at a cost. So, and the cost is, competitive ability or slower growth. So there comes a point when these two lines intersect. From then on, the resistant mutant has an advantage. And then comes the point when the resistant mutant goes through the zero line of growth. And in that window, there's selection for resistance. And you can see that if you make that curve steeper, the window actually gets more narrow, right? So what we did is we measured experimentally the steepness of pharmacodynamic curves, and on the left you see it for antimicrobial peptides, and then the rest is antibiotics from our own study and from other studies from the literature. And you can see immediately antimicrobial peptides, we measured 6 different ones and their combinations, have much steeper pharmacodynamic curves. And I should point out that the three-way interactions are always here. So two-way interactions and single drugs, they don't differ that much, but the three-way interactions actually improve the effect. So they make the curve steeper, and that should result in slower resistance evolution, lower probability, and it takes longer. Another important aspect about antimicrobial peptides, because of their killing mechanism, they don't induce mutagenesis. And that's shown here. So we have a positive control on the left. So we have 2 reporters, Reg-A and RPOS. And what you can see is, this is a disc in the middle with a drug on that. So the drug diffuses into the agar and you get a really clear area. So that's where all bacteria are killed. And then you get this beautiful halo, especially here with Reg-A, but you get also a halo here with that reporter gene. So that's an area where the bacteria are surviving, but they're stressed. And these are reporters of stress pathways. And these stress pathways lead to repair of DNA, and the polymerase that's used has a higher error rate than a normal one. So you get increased mutageneity. So if you look at, we have here 5 different antimicrobial peptides and lysozyme, and you see this is cecropin, the one that was discovered in 1981. Here's the human one. you have a very nice clear line, right? So bacteria are either alive or dead, they're never stressed. So it's basically like shooting somebody, they're just dead immediately. And therefore they don't elevate mutagenesis. And so if you, plot this as mutation rate per nucleotide, you see the control here. You see mutagenesis induced by ampicillin, ciprofloxacin, and kanamycin, and you see the antimicrobial peptides by comparison. Not all antibiotics increase mutagenesis, quite a lot. And that's something you can't avoid because of their killing mechanism. So then, together with Roland and his PhD student Desiree Beda at the time, we developed a model where we combined the pharmacodynamics with some genetics. And this is just a summary of the model output. So the x-axis shows you the drug concentration in terms of times MIC. So the MIC is here at 1.0. And at the top, we have an antibiotic. So that has all the traits of a conventional antibiotic we could measure. And at the bottom, we have an antimicrobial peptide. And you see just from the amount of color that the probability of resistance evolution is much lower for antimicrobial peptides than for antibiotics. And especially if you're above MIC, it's very hard for bacteria to evolve resistance. And we have here the mutation rate in there, the width of the mutant selection window, which we measure through this parameter kappa, so the shape of the pharmacodynamic curve, and we have fast killing, which is the difference in terms of time it takes to kill. And so that's quite reassuring, I think, and that seems to work quite well. And so this is just replotting data from Csaba Pál's lab in Hungary. So they did a big experiment with E. coli and exposed them to almost 20 different antibiotics and antimicrobial peptides. And so we've replotted it, it's just in a bar graph, and that's the MIC for change in resistance. So what you see is you get against antimicrobial peptides, you get resistance evolution, but on average it's 5 to 10-fold, while with antibiotics it's about 100-fold. That has to do with the possible resistance mechanisms that bacteria can actually evolve. Good, so resistance evolution against antimicrobial peptides is certainly harder than against conventional antibiotics. And there are more data out than the ones I show now. There are other labs working on that. There are data out from polymyxin as well, which has been used in the clinic, and that's all consistent with that notion that the probability of resistance evolution is really lower. But I just want to go back to one issue. So when we use drugs in human medicine, in most cases, as I said previously, we use one drug against one infection. And I won't go into that very much today, but I do a lot of work on insect immunity. And so AMPs are one of the main immune effectors. And one thing which is really different from what we do in medicine is they always use cocktails. They express cocktails of antimicrobial peptides, and they express antimicrobial peptides that in isolation are actually not active against the bacterium that infects them. Which to me suggests that there are actually a lot of interactions between antimicrobial peptides which we don't understand yet. And to make the kind of combinations even more extreme, I had a collaboration with Zvi Hayuka from Jerusalem. And Zvi is a chemist and he produces random peptides. So what he does is he uses 2 amino acids, lysine and phenylalanine, and then he defines the length of the molecule and synthesizes all combinations. So if you synthesize a length of 20 amino acid residues, you get about 1 million combinations. So this is really the extreme of combinations. And he's done this for a few years, and every time he produces a new batch, it has the same activity. And the kappa value, so the steepness of the curve, is the steepest curve we've ever measured with any antimicrobial peptide. So we did an experimental evolution approach. So, and as I said, that's relatively simple. You take a 96-well plate, expose them to 0.25 times the MIC, half the MIC, then you monitor when they regrow, then you increase the dosage, and you do this over weeks. So the experiment we did here with Pseudomonas aeruginosa, and we did the exposure for 4 weeks because that's the normal treatment duration, which is used for treating Pseudomonas infections in lungs of human patients. And so the postdoc who did that, Bernardo, did a lot of experimental evolution. I show you one set of those data here. So we have several different single antimicrobial peptides on the left, and then we have 3 random peptides on the right. FDK5, so they are 5-mer peptides, so the length of 5 amino acids. And then FK20 is 20 amino acids. So that's the one with the amazing number of combinations. And what you can see is that there is resistance evolves against all these antimicrobial peptides over the course of 4 weeks, not to a high degree, but it does. But it doesn't really to these these random peptides. And then we looked into cross-resistance. And that again goes back to what Michael said, there is this concern that there's a lot of cross-resistance. What you see on the left are actually the strains that are evolved either against melittin, hexagonins, cecropin, and so forth. And when we expose them to other antimicrobial peptides, when it's red, we have cross-resistance. So you can see that cross-resistance is actually quite a prevalent result, right, the outcome. The only difference is when it's green, it's collateral sensitivity. So being resistant to an antimicrobial peptide makes you more sensitive to that drug, and that's FK20, so that's the random peptide. So this is a bit of a wonder drug, that we don't claim that resistance can't evolve. We just think it's very, very hard. Because, as I said, I think bacteria can evolve resistance against everything. This will be a nightmare. I mean, that's not my problem, that's Zika's problem. He develops this as a drug. Get any regulator to convince them, because technically you would need to test every single combination and show it's not toxic, which with 1 million is always, it's obviously impossible. But there is, a way out to that. So random peptides make resistance evolution even harder. And I should point out that in nature, we don't have proper random peptides, but there are, for example, peptides in our own eyes, which are cleaved at random. So they have a combination of fragments, and that seems to change, with every time they're expressed. So that's also a way of generating in a natural system more than just having defined cocktails of defined peptides, but have cocktails of defined actually peptide residues. Okay, then I want to move on to responses to sublethal concentrations. And why are they important? So if you think about antimicrobials in the environment, and that can be in the soil, it can be in a gut, it can be in a biofilm, antimicrobials are at different concentrations. And quite often they're sublethal. And you have gradients, even in a natural situation where you, for example, have an organism expressing antimicrobial peptides to fend off its competitors, if you go away a bit, the concentration drops. So being exposed to sublethal concentrations, I think, is a reasonable assumption, and the same applies in medicine, because it depends on the drug dosage and the pharmacokinetics of what the drug concentration actually in the bloodstream, for example, of a patient is. And ideally, it's set up that it never drops below the MIC, but when you look into studies of how people actually adhere to these treatment regimens, you know that bacteria are often exposed to below MIC concentrations. And just for that, I want to introduce 2 concepts, which probably are familiar to some of you, but maybe not to everybody. So we have here a resistant strain, which would be defined by an increased MIC. And then we have susceptible ones, so that's the normal situation. But we also have 2 more types. We have tolerant cells and persistent cells. So they have the same MIC, but they show 2 effects. And the tolerant one is here. So you see that actually this is time here on the x-axis, and that's the bacterial death. And the susceptible ones die off, if you expose them to drugs. And the tolerant ones, they die off as well, but at a slower rate. So if you remove the treatment, too early, or because if the drug doesn't have a long half-life, then you have more cells if you have, scenarios tolerant cells. And then the other type are persister cells. And so that's a subpopulation of cells which is usually, people would estimate 1 in 10,000 cells. It depends on the combination between drug and bug. Can be 1 in 1,000, 1 in 100,000. And they kind of die off, but at a very slow rate. They just hang on. And I don't want to go into detail. I think by now more than 70 different mechanisms in different bacteria have been described of how persisters can form and what the genetic basis of it is. But I just want to look at the phenotype here. And so to start with, we did a simple experiment. We used pexigenin. So this is the first antimicrobial peptide from a multicellular organism that was developed into a drug by Michael Zaslow, and it's from the African clawed frog. And so we did an experiment. We exposed bacteria for 15 minutes to a concentration 1/10 of the MIC, then washed them, and then 2 hours later exposed them to 10 times the MIC. And these are the controls here. So that's just the killing phase. So what we find is two things. So we have the orange line are the ones that are primed, so they have seen a small concentration of antimicrobial peptide before, and the blue ones are the control. And what you can see is we get more, they get more tolerant, right? And we get more persisters. We get a biphasic response with persisters anyway, but we get more persisters if we prime the cell. So prior exposure does something with the cells. And then the paper that we described, we have candidate mechanisms of why that works, and that example with E. coli, I don't want to delve into that. But one thing which struck us is that if you have, if you switch on an immune system, you start expressing antimicrobial peptides, they enter the bloodstream or in the insect, the hemolymph, so the concentration increases. The same happens if you treat a patient with a drug, and then it's called pharmacokinetics, the drug concentration increases in the plasma, for example. So in every case where in any of these situations, bacteria at the start are actually exposed to a sublethal concentration. And then at some point, the concentration reaches MIC or 10 times MIC, and then we talk about killing. So we thought, if that's true, that, there's a very common situation where you're exposed first to sublethal concentrations, can we actually investigate it? And the other thing we looked into is resistance evolution. And I just showed two, this is a modeling approach with Desiree and Roland. Resistance evolution with primed and persister cells. And what we see here is the black line is controls and the blue is persisters. And so we have the probability of resistance evolution here and treatment intensity. And you can see that the persisters themselves in that scenario are not sufficient to actually increase resistance evolution. There's no difference. I have to say in the literature, there are some other data, especially from Nathalie Balaban's lab, which show that persisters can actually drive resistance evolution. In our example, we couldn't find that, but it's, it's our study. And then we looked into this priming effect, and that was very, very strong. So this prior treatment with 1/10 of the MIC actually, made resistance evolution much more likely. So what we did then is we did a simple experiment with small chemostats, which are shown here in the incubator. And we used precision pumps to increase the drug concentration in 3 different scenarios. So we have the time here on the x-axis and concentration on the y-axis, and we have a fast treatment where we reach within 90 seconds the MIC. Then we have an intermediate one where it takes 4 minutes, and a slow one where it's taking 15 minutes. So that's kind of taking the priming, but not having 1 dose of priming removing the drug, but just increasing it until we hit the MIC. And when we do that, we first look at the population size, and so the top one is the slower one, and we can see that over 2 hours, nothing really happens, and then bacteria start dying a bit. Priming is in red, blue is non-primed, so there's no effect of priming, which surprised us because that's what we previously with a different setup observed. But and then one thing we see here is intermediate concentration, fast concentration, so you get faster killing, and clearance of the population if you increase the drug concentration faster. So that's all kind of to be expected. But what we were really interested in were the number of persisters. And so the top graph shows the persisters after 15 minutes, for slow on the left, intermediate in the middle, and fast on the right. And you see that already there's there are more persisters in the slow ones, and the effect gets more pronounced after 60 minutes. We can't measure it anymore in the fast treatment because there's nothing left. And then after 120 minutes, we only have persisters, and a quite a high number, an increased number over expectations on normal persistor ratios in the slow treatment. So that's just a simple proof of principle, but that pharmacokinetics at least can drive the proportion of persistor formation in a bacterial population. Whether that would play out in a more complex physiological environment like a human immune system, we don't know. But at least in the lab, we can see we can actually induce more persisters by playing a bit with the pharmacokinetics. Okay, I just want to sum up that session, that part of my talk on antimicrobial peptides, was pointing out that the pharmacodynamics of antimicrobial peptides and the fact that, as far as we know, most multicellular organisms deploy them really as combinations can be actually quite useful. Conventional antibiotics, even though resistance evolution is slower, we're still wasting a great opportunity. I think we should get informed by studying how AMPs are used in natural systems, where they've been shaped by natural selection and the combination has been shaped, to use them in a cleverer way so we have them for a longer time. Because developing drugs and getting drugs to the market is a very lengthy period and very expensive. And we think we are at the moment at the cusp of a development where we have antimicrobial peptides as new drug candidates. There's a great opportunity to deploy them differently. Okay, and with that, I want to move to the last part of the talk, forecasting resistance and whether we can use in vitro experiments. And so when we talk about resistance and what we do with it, there are two ways, or two different kind of problems. One is management. So we have resistant bacteria. What do we do with them? And the other one is prevention. No, there's no resistance yet. How do we actually prevent the evolution of resistance or delay it as long as possible? And the kind of standard three approaches that get used are prudent use. So we only really use antibiotics when we need them. New drugs, that's a problem because drug development is very expensive. Antimicrobials, in most cases, are used for 3 to 4 days these days. So there's not much money to be earned. So for the companies that invest, there's not a great opportunity for them to actually get their investment back. So they don't do it, or not very much. And the other one, which is developing very fast with microfluidic technologies, is rapid diagnostics. So really know which bacteria infect a person and to know whether they have any resistance so that you can use the right drug from the start and don't lose any time. And also, diagnosis more simple. Is it actually viral infection or an bacterial infection? So for example, during COVID, the use of antibiotics worldwide increased because people had symptoms of colds and a lot of doctors gave them antibiotics, and then there are countries where you can buy antibiotics over the counter. So that actually the sales figures shot up, and that obviously then can possibly drive resistance evolution. But what we don't do is forecasting. But I want to highlight one thing. So there is a correlation, and interesting enough, this is mostly in industrialized countries, between the usage of antibiotics. So green is Sweden, red is Greece, and blue is France. This is just for one drug, ciprofloxacin, over about 12 years. And you can see there a much higher dosage per head, per population in France and Greece than there's in Sweden. And that's perfectly mirrored in the proportion of resistant E. coli. So France and Greece have a much higher proportion of resistant E. coli than Sweden. And, I think that's probably very straightforward. But what I want to use this figure for is I want to point out, when you read about antibiotic resistance evolution, people always talk about misuse of drugs, overuse, wrong diagnostics. And even though I'm in France, I would call that kind of difference between the prescriptions and the resistance in Sweden and France and Greece misuse, because you can obviously do better if you have proper, have the appropriate public health policies, but still use causes antibiotic resistance. We should be aware of that. So the problem, even if we get much better and follow the countries who are really good, which is mostly Scandinavia and the Netherlands, they have the most restrictive policies on that, we still face the problem of antibiotic resistance evolution. It's non-escapable. So then I got together with a few colleagues, with Michael and then Sebastian and Roland, who are modelers, and then Charlotte is a pharmacologist, and Rasmus is actually a medical doctor who collects the antibiotic resistance data for the University Clinic, the Charité in Berlin, to propose the idea that in addition to what we do already, we should think more about whether we can actually forecast. So we have ideas about how much resistance is there, which drugs have we, can we inform future drug usage. And there are several ways to do it, and there have been papers, and we have a table with studies that have done it. I just picked two here. So there are ways of, we have actually surveillance data, we know how many, or what the proportion of resistant bacteria of a particular species is and which drugs have been used, and then we can make forecasts. One other thing, and that's what I kind of wrote most about in that opinion paper, is experimental evolution, because that's what I'm working on, and we are also with engineers trying to build a simple kind of evolution machine, which is a long-going project, and I don't know whether we'll ever succeed, but the idea is that you can have a simple machine where you can test how fast resistance evolves against a particular drug. And one criticism we face all the time, as I said at the start, and everybody I know in the field is, you do experimental evolution in vitro, and you do it, a simple way, or just with a pipette, or in a more smart way with a pipetting robot. There will always be a review and say, it's all fine, but it doesn't tell us anything what's going on. Wow. And, there are major differences. So in vitro, you can get resistance against most drugs within 1 to 2 weeks, even against, antimicrobial peptides, where it's very hard. And even this proposed wonder drug teixobactam, which was published to great acclaim 10 years ago, takes 4 weeks, and people get low-level resistance, but they get resistance. So in real life, it takes 2 years for most antibiotics. And I said colistin 6 years, polymyxin, which is basically related to colistin, takes 12 plus years. So there's a huge gap in time. So maybe that criticism is correct. So what we did is we did a systematic review with 2 postdocs and a PhD student in my lab to investigate how much these two match. So we in total started with 700 papers to screen, which had experimental evolution data in there. And then we had certain criteria. They needed to actually have an MIC, they needed to provide genetic data, so genomic data, so we know anything about the resistance mechanism. And after all that screening, there's 67 papers left, which are shown on the left, the black bar, and then, and because one paper can have more than one data set, yeah. So what you see on the right is not, some of that is more than the total, simply because of it. So anyway, what you see is most experimental evolution studies are on Pseudomonas, then it's E. coli, and then it's Staph aureus, and then it kind of drops off. All the 6 escape pathogens are covered by that, but there are really 3 pathogens where we have sufficient data. And these are the antimicrobials studied, and that's a wide variety of antimicrobials. The most popular one is ciprofloxacin, and interesting enough, quite a lot of AMPs and colistin as well. So it's not all myla because we don't work on colistin. And then you have an even distribution of lots of others, and most major groups of antibiotics are actually in this data set. And so when you look into this a bit more detail, we focused in on the three most abundant bacteria in these studies, E. coli, Pseudomonas aeruginosa, and Staph aureus. You see that actually, people study all these different antibiotics, but ciprofloxacin, which is the third one, really sticks out. Most studies in E. coli and Pseudomonas are actually done with ciprofloxacin. So we use that for further analysis. We looked also into this paper needed to provide MICs, so we can make some useful comparisons, and you see that in all these examples, the MICs increase. There's a fold in change on the left-hand side, so it's between 30, which is the lowest for colistin, not surprising, it's an antimicrobial peptide, and up to 100-fold for some others. So then the next step was to ask, that's fine, that's a phenotype, so you get a resistant phenotype in the lab, you get a resistant phenotype in patient strains and environmental strains from wastewater and soils and so forth. How does it look at the genetic level? And that's obviously, I have a personal pet hate for heat maps anyway, but what you can get from this, we have here 5 different antibiotics and all these amino acid changes. And what you can see is it's very specific to all these different drugs. And we get most changes for fluoroquinolone, ciprofloxacin, which is also a function of the fact that these are most data in our database. So how does that match really with patient data? That's the final step. And so these are here the main mutations, resistance mutations from the experimental strains. And the most abundant one is gyrase A, which is a very important resistance mechanism against ciprofloxacin. And then we have NFXB, PAR-C, gyrase B, PAR-E. So this is the kind of order it pops up in the experimental evolution studies. And the result is, for me, is very pleasing because that's what we get from patient strains. So the most important one is gyrase A. And then it's the same one as here, only PAR-C and NFXB swap places. So actually the matching in terms of what you get within a 2 to 4 week experiment in the lab in terms of resistance mutations you find in your simple in vitro experiments pretty much reflects what you find in patient strains. Now, we're still far away from forecasting, yeah? But at least to me that's very encouraging, and we hope to other people in the field when we publish that these in vitro experiments are not only interesting to study evolutionary questions, but they might actually have some applied value, and that there is the, the notion of doing resistance evolution in vitro to inform actually the use of antibiotics, for example, in the clinic is actually useful and is possible. It will be a long way, but it's nowhere near this criticism, oh, it doesn't tell us anything. It's actually the opposite. So this is only data for Pseudomonas and ciprofloxacin, I should say. The Ecology data we're currently analyzing, so I might be less optimistic once we've analyzed them, I don't know. But for the moment, I stay optimistic. That's the useful approach. And so we also looked into the MIC, and what's interesting, the NIC of the in vitro ones is for the particular resistance mutations is blue, and it's orange for the patient strains, and there's some difference. And that's because obviously people who do in vitro evolution, they use usually 1 of 2 strains of Pseudomonas, while the strains from the patients have all sorts of different genetic backgrounds. So, and obviously not every single resistance mutation shows the same phenotype or strength of phenotype, I should say, in a different genetic background. But that's not surprising, I don't think. Okay, and then one thing we proposed in our paper is that resistance could really be part of developing antibiotic stewardship plans. So what clinicians in most hospitals I know of do is they have plans and they are informed by learned societies to say you have that bacterium, then you use this drug. If that drug fails, you use the next one, and so forth. And they also consider things like the price, they consider toxicity, and so forth. And we think that we could combine that with also a resistance score. I know I was just learned last week, but I didn't have the opportunity to talk to these clinicians. There's actually a clinic in Switzerland which has introduced a traffic light system which includes probability of resistance evolution to inform their antibiotic stewardship. Okay, so to conclude my talk, resistance evolution against antimicrobial peptides is hard, and against antimicrobial peptide combinations is even harder. So that really supports the applied argument that we should use more drug combinations. Pharmacokinetics can help to reduce persisters, but there's still a debate, as exciting as the persister field is, how important persisters are really in driving resistance evolution. And that forecasting, I think it will be, a big endeavor to go towards forecasting resistance evolution as an additional tool to the measures we've got, but I think it's worthwhile following that. And I also think that, in vitro evolution can actually play a part of that. Okay, with that, I want to thank you and my funders. Thank you very much.
Thank you for the talk. I was thinking one thing where people, I guess, criticize the in vitro versus in vivo is that there's the whole immune system that we want to produce and stuff. And as you work on insects, I was wondering, is it something feasible now, or do you use it already to do in lab, having insects as hosts, and still being in the lab, still being able to maybe monitor some things? Like usually, I guess, the in-between is the small animal, mice, but it's not very easy to practice compared to an insect I would expect. So what do you think of this in-between strategy with it?
So we actually have an ongoing, but we don't have data yet. We have an ongoing experiment with gene knockdowns where we test in vivo resistance evolution in insects.
Does it make it more easy to do it in insects than to mice, or is it just as complicated?
Well, I think it's easier. Well, first of all, the sample size is not limited. And then the other thing is you have no acquired immune system to consider. So you just have the immune system, which is based on phagocytosis, oxidizing enzymes, and antimicrobial peptides. And you can reduce the complexity of antimicrobial peptides by gene knockdowns. I mean, you can do the gene knockdowns in mice, but getting a sufficient sample size for an in vivo resistance evolution study is possible, and there are very few. Andrew Reed has done stuff on Plasmodium, but it's very limited what you can do. But it's not, utilized to the extent it probably should be.
Yes, thank you for the talk. I was wondering if you tried applying those AMPs in more complex biological ecosystem or microbial ecosystem where you have multiple strains or multiple species of bacteria, because I guess it's one big difference also between the in vitro and real life.
No, we haven't done that yet. I don't know of any published study that's done it in a really complex environment. I mean, lots with antibiotics, but I don't think there's anyone with antimicrobial peptides. But I have a colleague who's a soil ecologist who recently became interested in antimicrobial resistance, so I hope he can take that on.
Thanks for a super interesting talk. A lot of your work is focused on the evolution of resistance, right? New resistant strains. But there are some examples, for example, malaria, where resistance has only evolved a handful of times across the world, and yet, all Plasmodium, now carry some of these resistance alleles that are apparently really hard to evolve, but once they're there, they can spread widely. And I wonder if you have, if you're thinking about predicting that as well, in addition to just, the evolution of new resistant strains.
I mean, that would be very hard. So I think one needs to do the first steps first. I mean, for colistin resistance, it's true. This is MCR1 gene, and that was published 10 years ago, so that's been probably around for 15 years or whatever, and that has spread. So the first study was published in China, the next one was been published a year later in Germany and was the same mutation, which strongly suggests that that's has been transferred, because it's a big industry and there's a lot of exchange. It would be possible because there's only very few solutions to become colistin resistant. That's parallel evolution, which you can't exclude, but it's much less likely. But yeah, I mean, we discussed it yesterday, so it really depends on bacterial species whether resistance mechanism spreads globally or evolves locally. For Staph aureus, for example, we know that there are strains which actually have a global spread. Yeah, that will complicate things. And that's a bridge we cross when we get there.
And then an internet.
Can you put your slide back on the in vitro versus in vivo or resistance mutations?
This one?
Yeah, this one. So there's still a little bit of a discrepancy between quality. like for the NFX.
Yeah, so they are in different position, par C and NFX B. Yeah.
And from my experience, at least in vitro in the lab, you can somehow, especially among those mutations, tune the distribution of the frequency according to the dose of antibiotics that you will be using in vitro. And so you can make it almost as you like, more or less. So is it just chance here, or because you need to choose a dose, right? And so what do you do to really reflect on? Imagine you'd like to make some real prediction. So which dose would you use?
Yeah. So this is kind of haphazard, right? So these are individual labs who, for whatever reason, chose a particular approach with a particular drug and a particular dosage rate.
Yeah.
The same is true for these patient strains. This is not a systematic surveillance. This is labs who, or, hospitals which had the opportunity to sequence stuff. And not all sequence stuff from hospitals is actually available in a public database. So we can only obviously access public database data. So that's the match we get. But I fully agree. The next challenge is then, if one wants to use that for prediction, to think about in the experimental evolution setup, how you actually set up your pharmacokinetics, and possibly also what medium you use to make it fit even better than what we find here by these kind of random studies, which are basically from the perspective of that question. And that will be the next step. And you can do some of this stuff with the pipetting robot, where you have rather complex pharmacokinetics. And that's very useful from a scientific point of view, but that will never be used by any hospital. Because those people who have a pipetting robot, they have usually dedicated staff who do nothing else than running this robot, and no hospital has extra staff. And that's why we try to have, build a little reactor which kind of mimics physiological changes and concentration gradients. So we're working on that, and we get concentration gradients established, and that all works. And now the next step is to make that repeatable and have it sterile, and there are all these kind of practical challenges which we need to overcome.
Just very quickly, following on from Thomas' question, can you do a statistical model where you add dose, if it's known for the studies, and look at the...
For these studies?
Yeah, and then look at the rank.
We have the dose for most of those, but we have none of the doses for that.
You don't know at all?
No.
Okay.
So for those data, it's impossible, unfortunately.
Okay.
Yeah, we have two questions online. So first question by Christophe Renaud. Christophe, if you want to open your mic, you can, and ask your question directly.
Yeah, hello. Thanks, great talk. Thank you very much. You have shown that about a number of bacteria produce antimicrobial peptides. And I was wondering how they protect themselves against their own AMP, and if the mechanism would be known, if this could inform us about general mechanisms of resistance.
Okay, so the general mechanisms of... You need to switch off your microphone. So the general resistance mechanisms are really changes to the cell membrane. So that can be either by reducing the charge or adding, for example, molecules that physically hinder the attachment of the AMPs. But when you have a bacterium that's producing it, there are different ways of doing that. It could do it in an inactive form, which only gets cleaved once it's outside, or it could be transported then in a membrane vesicle, which then, yeah.
Immunity proteins are produced
Yeah.
together.
Exactly. So, and then it only becomes active when it's outside.
All right, and we have a second question by Rémi Froissart. Rémi, you should be able to open your mic now.
Yes, do you hear me?
Yeah,
perfect.
Thank you very much for the presentation, very interesting. I'm working on phage therapy, so usually I'm supposed to be in Montpellier, but now I had to go on a PhD defense. So I'm not there, unfortunately, because I would be very interested to meet you. So knowing that bacteriophages and bacteria are co-evolving since billions of years, and bacteria becoming resistant to phages, would you consider, based on your work, that phage therapy is not a good alternative or complement to antibiotherapy?
Well, actually, I think that Michael would be in a much better position to answer that, because my knowledge about phages is quite limited. I know of a few studies which combine antibiotics with phages, but I don't know a study which combines antimicrobial peptides with phages, which would be interesting in itself. But with phage therapy, that would be pushing out the boat. I can't really say anything on that.
Yeah, but based on your knowledge, well, all your approach is based on the resistance, right? So if we use cocktails, then we should decrease the probability of seeing any resistant bacteria. So somehow it's based on my question is a bit based on the slope.
Okay.
That you are talking about, and of seeing the seeing resistant bacteria to appear. No, I should talk more.
Yeah, no, that's certainly true. So from what we have measured so far, if you use a combination, you always make the slope of this pharmacodynamic function steeper. I didn't put the data in, but we've measured that also for these random peptides. And the steeper the slope is, the smaller is the mutant selection window. And so that kind of model approach is consistent with all the experimental data we know of, and the few data we have from polymyxin, colistin. And some people would even claim daptomycin, which is an peptide antibiotic, but which basically has a very similar function. So it is membrane active.
Okay, last 2 more questions. Sylvain, then Delphine.
Okay, so a question about the first part of your talk, you mentioned that there's no stress when you apply AMP, and then later on you talk about priming, and I was wondering how you can reconcile the fact that maybe with certain concentration of antimicrobial peptide, maybe, obviously the bacteria change, so I was...
Okay, so I wasn't very clear about this. So the stress we measure with these reporters about mutagenesis, these are stress reporters, and these are stress responses which elicit DNA repair. And what the cells do in the priming experiment, for example, they put more cholic acid on the surface, and that kind of stops the AMP from interacting. So they show a physiological response, but these kind of canonical stress pathways, which have mostly to do with intracellular activities, they are not switched on. So that was a bit sloppy, sorry.
Yes, thank you for a nice talk. My question is about the choice you made to select antimicrobial peptides. Most of them are membrane active. And so how much of the difference you see between antimicrobial peptides and antibiotics is due to this choice? In other words, if you had compared vancomycin to peptazine, would you see differences in the pharmacokinetics and in the mutant rate observed? Because they have the same target, precursor of the peptidoglycan, so they are involved the same target. And we know a bunch of antibiotics that do the same, and a bunch of antimicrobial peptides that do the same. So you could compare these 2 sets, and maybe you would see less difference between the 2 groups. Yes, I have.
That is probably true. We haven't measured that. I mean, and the ones with intracellular targets, all the protein-rich ones certainly will have a different killing dynamic.
As you said, most of them are membrane active.
Exactly, so that's why we did that.
Far from that.
And to be honest, we also, we did it for pragmatic reason, because synthesizing AMPs is very expensive.
I know.
So you get the ones you can get hold of. And pexigenium I can get from Michael for free. No, I mean, that's also that I have to say, you know, if they are going to the market, they're very costly and they need to be modified because the synthesis is very complex and that drives the cost for them. And that's why so far they haven't been considered as being attractive. But as you see on the preclinical list, because we're running out of other substances, that's why people now turn to AMPs. And then these technical issues have to be resolved. The easy thing for me as an evolutionary biologist who has nothing to do with drug development, I can say it's not my problem, but I have to admit it's a huge problem.
Just, I was wondering, do people are concerned also about the potential side effects for our cells? Can antibacterial peptides harm our cells?
They are, which I think in a way it's important, in the other way it's funny, because when you talk to these people, they make the assumption that antibiotics don't have any side effects. And it's certainly true that all antibiotics have side effects. To start with, they mess up your gut microbiota. Ciprofloxacin, for example, which where the usage has dropped significantly in most European countries, has quite significant implications, for example, for your Achilles tendon. So people actually rupture their Achilles tendon, you know, and the probability is much more increased if you take ciprofloxacin. I think any drug will have side effects, but as a kind of principal argument against, you know, this new group, I don't think that's a valid argument. The other question, which Michael, and there also came out another question, which I want to, you know, finish off with. We've done a study. So, is there cross-resistance to our own innate immune defense if you use antimicrobial peptides? Because the peptides are conserved, the resistance mechanisms in the bacteria are conserved and limited. The only study who's tested that with bacteria that are resistant against a particular host antimicrobial peptides is a study we published in beetles. And this gets studied as evidence that there is no such problem. I find it a bit irritating, so, you know, to rely on a beetle study on one to make that claim. And that's certainly something people who work on, you know, vertebrate AMPs, and especially people who develop antimicrobial peptide drugs